Hi, and welcome back to Intro to Biology for Majors, or BIOL 1224. This is going to cover Chapter 11, which is from your Openstax Biology 2E book. Chapter 11 covers meiosis and sexual reproduction, and this should come from Module 3 of our course. So we're starting off with our objectives, and there are 3 for this chapter. The first being recognize the role of meiosis, particularly in humans. The second is recognize the role of crossover, random alignment, and random fertilization in variability. And the third is identify potential sources of error in meiosis. So let's start off by talking about sexual reproduction. So in sexual reproduction, the DNA of the offspring comes from 2 parents. A female parent contributes an egg and a male parent contributes sperm. These cells fuse during fertilization, yielding the first cell, or what we call a zygote, of the next generation. Sexual reproduction produces genetic diversity among offspring, which increases the chance that some offspring survive in changing environments. So if you look at the image on the right side of the screen, I have an image of a female, which would be the mother, and a male, which would be the father. The female produces an egg, the male produces sperm. They come together at the site of fertilization and they make that zygote that we talked about. So they have a total of 46 chromosomes that are in 23 pairs. So 23 come from mom, 23 come from dad. We have 46 chromosomes. And then that goes together to make our embryo, which once again, we have 46 chromosomes and 23 pairs. So this happens, and what we said here is sexual reproduction produces genetic diversity. So this genetic diversity basically is trying to say this is why you don't look exactly like your siblings. It's why there is diversity among family units themselves. So in the animal world, it's a good thing because whenever you have diversity among offspring, it's a higher chance that some offspring are going to survive to adulthood. So if some offspring have environmental or physical issues that don't allow them to survive to adulthood, there should be others that allow that to happen. So once again, the final products of meiosis in humans are gametes. So we talked about meiosis a little bit in the previous slideshow. The gametes I'm talking about are sperm produced by the males or egg produced by the females. And cloning is what we talked about in the previous slideshow, where we have an asexual reproduction. So that's the opposite of what we're talking about here. It's commonly used by bacteria. We talked about that in the previous slideshow as well. So let's talk about diploid cells. And in our previous slideshow, we actually talked about both haploid and diploid cells. So our haploid cells we talked about were our gamete cells, our germ cells, the sperm and the egg. So haploid means what we call 1 in. So there's an image here that kind of tells you what I'm talking about. Haploid would be 1 in. So there's only 1 set of those chromosomes that we're talking about with the sperm and 1 set with the egg. So most cells in a sexually reproducing organism actually have 2 sets of chromosomes, which would make them diploid, like a zygote. So in that previous slide that we saw, we come up with the zygote that makes the embryo. That zygote is 2 in because it has chromosomes from both parents, from the sperm and the egg. So that's a 2 in diploid zygote. So 1 from the father, 1 from the mother. Cells with 2 sets of chromosomes are called diploid cells, or what we call 2 in. So then we talked about the process of mitosis, which leads to a fetus, which leads to an infant, all of which are 2 in. They have the 2 sets of chromosomes from both parents. An adolescent, an adult, we undergo meiosis. Here we have the process starting over, and we start off with sperm from father, egg from mother, that are each 1 in, and then they go to a 2 in zygote. So that's the process of creating diploid cells. So now let's talk about autosomes. So this image here breaks down our chromosomes. So like we said in previous slides, we have 46 total. We have 23 pairs. So this purple box here shows you 22 of those 23 pairs. And those 22 are actually autosomes. It starts in the sentence here, but it says the numbered chromosomes that you see in purple here are called autosomes, and they're actually the same for both sexes. So they're the same for male and female. There is a link in the notes section of this if you really want to get into a karyotype and kind of what each chromosome is paired with and how they work together. But the 2 letter chromosomes that you see down here at the bottom, they're the sex chromosomes. So these are what determine male versus female. So in humans, an XX chromosome is going to be female and an XY is going to be male. And the male karyotype is what's shown in the image whenever you go to this link if you want to see that. So yeah, humans have 22 sets of autosomal chromosomes, which are all in purple. They're the same size and shape in both sexes, and humans have one pair of sex chromosomes, X and Y. So once again, XY would be for male, and XX would be for female. X and Y are not the same size and shape, so that's the only one that really differs between both genders. So let's talk a little bit more about a zygote. So the image here on the left side of the screen I put together kind of shows you what we've talked about previously. So we talked about haploid, meaning 1N, which means it contains one set of chromosomes, and this would be a sperm. So this is from the male. This is then representing an egg. So this is a haploid, or 1N, containing one set of chromosomes from the female. And then down here is our zygote, which we're talking about in this slide, which is now diploid because it has genetic material from both male and female, from both mom and dad. So gametes, like sperm and egg cells, are haploid sex cells. A haploid cell has only one set of chromosomes. When two haploid cells fuse at fertilization, one diploid zygote is formed. A zygote is the first cell of a new organism. So we did see that in a previous slide, but I just want to hit on it again and kind of reintroduce what a zygote is. So a zygote is the first cell of the new organism. So now I'd like to talk about meiosis. And in the next slide, I am going to talk about mitosis versus meiosis. So I'm going to break down the difference between the two, because in the previous slideshow of chapter 14 and 15, we talked more about the process of mitosis. And in this slideshow with sexual reproduction, we're talking more about meiosis. So I do want you to keep in mind there are two separate processes that come up with separate cells, and there are different reasons that our bodies do each of them. So keep that in mind, and if you need a breakdown, I'll do that in the next slide. But let's talk a little bit about meiosis and its purpose. So in the process of meiosis, DNA will replicate once, but the nucleus will actually divide twice. So there are 2 rounds of what we call nuclear division, which results in actually 4 new nuclei, and usually 4 daughter cells, each with half the number of chromosomes as the parent cell. So previously, when we spoke about the process of mitosis, we were actually cloning cells. We were making exact clones of the parent cell in these new daughter cells. So meiosis is different in that each of the new cells has only 1/2 the number of chromosomes as the parent. So it is not a clone. It only has half the chromosomes. The other big difference between meiosis and mitosis is that mitosis, which we previously talked about, comes up with 2 daughter cells. That's what you end up with. So you end up with one exact clone of the parent. But the process of meiosis, you actually end up with 4 daughter cells, and each of them only has half the number of chromosomes as the parent. So they're not clones. So the first in meiosis separates homologs, the first process of nuclear division, and then the second process of nuclear division separates chromatids into individual chromosomes. So we're going to look more into that in the next few slides, but there are some points in the notes that I want to either hit again or just kind of talk more about. So once again, meiosis is used for actually just one purpose in the human body, and that purpose is the production of gametes, what we call sex cells or sperm and egg. So the process of meiosis in the human body is only for creating sex cells. And we did talk about mitosis previously. The process of mitosis is for things like wound healing, cell replication. But once again, meiosis is just for creating sex cells. Its goal is to make daughter cells with exactly half as many chromosomes as the starting cell. Meiosis will successfully result in 4 different haploid cells. So we're going to talk a little bit about mitosis here. So the difference between meiosis and mitosis. Mitosis will actually generate body or somatic cells, which we talked about previously. They are needed for wound healing. It is for meiosis, once again, is for generating gametes. So mitosis, body cells, wound healing, meiosis, generating gametes, sex cells. So chromosomes replicate during the process of interphase or S phase, and crossover occurs in prophase. And in this instance, we're talking about meiosis. So in meiosis, chromosomes will actually condense during prophase, and cytokinesis is the final step that follows both mitosis and meiosis. So the processes themselves, you'll see in the next slide, they look similar until you get towards the end and you realize that you're going to come up with only 2 clone cells in mitosis, and in meiosis you're going to come up with those 4 daughter cells that only have half of the chromosomes as the parent. So the reason that you are different from your siblings is that cells produced in meiosis are different from one another. So 3 events make that happen, and those events are what we call crossover, random alignment, and random fertilization. And we're going to look more into those in the next few slides. But once again, this is that genetic diversity that we talked about in the beginning of this PowerPoint. So the reason that we are different from our siblings, even most of us being genetically different from twins, is that our meiosis process creates 4 new cells that only have half the chromosomes, and they're all different from one another. Okay, so I'm hoping that this slide will really kind of break down the differences between mitosis, which we talked about in previous slides, and meiosis, which this slideshow is really talking about. So once again, mitosis, which would be right here on the left, this up vertically, you can see the whole process starting at the top, ending at the bottom. Mitosis creates somatic cells, which are body cells. And the instance that they give for this is an example of wound healing. So that is the process of mitosis. So let's go ahead and go through it because we have talked about mitosis previously, but I want you to really understand the difference. So you're starting off with a diploid or 2N parent cell, and then chromosome replication takes place during prophase. Prophase here, we have chromosomes that condense and become visible. So these are the chromosomes inside. So you'll see that you have a blue and a red. You still have blue and red down here, but in this prophase, we actually have chromosomes condensing and becoming visible. Then we go to metaphase, where the chromosomes line up across the middle of the cell. And then we go into anaphase and telophase, where chromosomes are actually pulled towards the opposite poles, and the poles condense. And the end result of the process of mitosis is 2 diploid daughter cells that come from this process. And these diploid cells are exact clones of this parent. So they look the exact same, they have the exact number of chromosomes, they look the same, they work the same. That's the process of mitosis. And then these cells go towards things like wound healing, they are body cells. So now let's forget about mitosis for a second and talk more about meiosis, which is what's very important for this slideshow because this is very important for the process of sexual reproduction. So the process of meiosis starts off with a parent cell that's diploid. So it has 2N, it has both female and male chromosomes. And then we end up going to chromosome replication, similar to mitosis. We go into prophase 1, similar to mitosis, where in prophase 1 we have chromosomes condense and becomes visible. Often what we call crossover will take place in prophase 1, so we'll look into crossover in the next slide. Then we have metaphase 1, so chromosomes are lined up. And as you can see, there's a slight difference whenever you look at these chromosomes now. So the process of crossover, which we'll talk about next, changes these chromosomes. So we don't just have lined up blue and red chromosomes anymore. Now we have chromosomes that have a little bit of blue and a little bit of red on either side. So they're mixed together rather than being completely separate like they are in mitosis. So that happens in meiosis. It kind of switches up our chromosomes rather than keeping them identical. So in metaphase 1, once again, chromosomes are lined up and 2 diploid daughter cells are formed. So now we have 2 diploid daughter cells here. And then what happens in anaphase and telophase 1, the chromosomes are pulled towards the opposite poles and they decondense. And now in the process of meiosis, we have 4 haploid, what we call 1N daughter cells that are formed. So the end result is 1, 2, 3, 4 daughter cells that are 2N, okay? That are 1N. And technically, if you want to look at daughter cells as 2N, you can have 2 here and 2 here. But yeah, the process of meiosis leads to 4 one-in daughter cells. So now let's look deeper into the process of crossover. So during crossing over, 2 homologous chromosomes will actually exchange genetic material. So if you go back to the previous slide and look at what it looked like, in the process of mitosis, we just have metaphase, where the chromosomes will line up across the middle of the cell. But in meiosis, which is what we really want to talk about here, we have metaphase 1, and in that, we actually have the process of crossing over. And so whenever we talked about that genetic diversity and how these daughter cells are all different, the process of crossing over is extremely important to that. So crossover occurs in meiosis, actually during prophase 1. Crossing over is very common. Multiple crossovers will actually take place for each pair of chromosomes, and it's no more likely in males than in female gametes. So it happens randomly for both male and female gametes. Prophase 1 is the only phase of meiosis with exchanging of genetic material between 2 chromosomes. So during crossing over, once again, the 2 homologous chromosomes exchange genetic material. So let's look at this. So we have homologous chromosomes, which basically means all, let's just use blue for male and red for female. So in this example, we have all male chromosomes homologous here, all female chromosomes homologous here. So homologous just means the whole thing is all male, and then in this case, the whole thing is all female. So let's look at the process of chromosome crossover. So literally what it looks like is the male chromosome and female chromosome literally cross over one another physically. So when they do that, they have what we call recombinant chromatids. So whenever I think of the word recombinant, I always think of the word recombination. So now there's a new combination of chromatids within these chromosomes. So now the male has a little bit of female and the female has a little bit of male. And its non-recombinant chromatids are on the sides that are still homologous, right? So this side is still all blue, this side is still all red. But once again, in the middle here, we have those recombinant chromatids that have changed. So we have the male with a little bit of female genetic material, the female with a little bit of male genetic material. And once again, whenever we now have these 4 daughter cells that are going to come out of this process, only 2 of them are going to have this recombinant DNA, and the other 2 are going to be the non-recombinant chromatids. But we're still ending up with 4 daughter cells that are all genetically different from one another. So meiosis actually generates enormous variability. And when we're talking about variability, I mean within our genetic sequence, within our chromosomes, which once again is why we are all different than our siblings, than our cousins, than our parents. So let's kind of look at the processes that really cause that variability. So we've already talked about crossing over, which I have a photo right here. Basically, we have homologous chromosomes still present within this cell. So we have, if we're using male and female as red female and blue male, these would be all female and all male chromosomes. We also have what we have here, the crossing over that we spoke of in the last slide. So basically where these chromosomes are touching, they're going to be crossing over, and then we'll have some recombinant DNA found here. So that's crossover, and once again, that's going to cause a genetic variability because we have 4 daughter cells coming out that are all different. This happens during the process of meiosis, and crossing over helps us to really get that recombinant DNA going and a change within each of those daughter cells that could then lead to different physical or genetic material moving on to the offspring. So once again, the gametes produced during meiosis are different from one another because of crossing over, which we just talked about, and because of random alignment, which happens in metaphase 1. So in the process of mitosis, alignment is actually very perfected and sought out in a very certain way. But in meiosis, we have a more random alignment that happens within metaphase. And so especially after crossing over, where we have all this recombinant genetic material, when our chromosomes line up, they're not going to be pulled apart exactly the same on both sides like they would be in the process of mitosis. We're going to have random male and female genetic material being pulled in either direction. So we're making those 4 different daughter cells rather than cells that look exactly like the parent. So I want to dig a little bit more into random alignment. So random alignment pairs chromosomes randomly during the process of metaphase 1. In humans, there are actually over 8 million possible combinations of paternal and maternal chromosomes, which once again is why even in 8 million children from the same two parents, none of them are going to come out the same. So we have this process that basically lines up the chromosomes, but does it in more of a random way than it would happen in something like mitosis, where you have exact copies of parent cells. So this random alignment helps with that genetic variability that we're going for in meiosis. So this is a very cool slide to look at. Basically, this is going to talk about the variability in twins. So whenever I was talking about how we all look different, we all have different physical and genetic traits from our parents, often we do have twins. So we do have people that may look the exact same, but genetically they are actually not as identical as you may think. So if we're looking at this image, on part A, I actually have fraternal twins. So basically, this whole slide is going to show us the possible relations of fetal membranes of what we call monozygotic twins. So basically, we're going to talk about fraternal twins in two different ways, and then also identical twins. So the meaning of fraternal twins, if you don't know, fraternal twins don't physically look the same. And often it would be one male and one female that come from the same egg or that are born at the same time. But identical twins are the same sex, and they look physically identical to one another. So let's break down fraternal twins. So both A and B are going to show examples of having fraternal twins. So in part A, we have splitting that occurs at the 2-cell stage, and each embryo has its own placenta, amniotic activity, and chorionic activity. So basically, we have the initial zygote blastocyst here. So we have basically what's going to be one baby on this side, what's going to be the other baby on this side. And then we go through the process here, and we now have two separate placentas that are within the mother. So that's the sac that holds the baby. There's also an amniotic cavity here. They're both separate, so the babies form basically in completely separate placentas and completely separate cavities. And if you look here, you can see a more generalized image of what that'll look like. So there's two different placentas, which are represented here in orange. And then you can see that the fetus or the babies in blue right here, they're technically within the same mother at the same time, but they're not necessarily going to look or be genetically alike in any way, because these actually did not come from the same egg. So these are two separate eggs, two separate sperms that then live in two separate placentas and are born within a few minutes of each other, but don't physically or genetically have to resemble each other. So that's one way of having fraternal twins. There is a second way. So you can have fraternal twins that actually do come from the same egg. So if you look at this example, we have an inner cell mass on this side and another inner cell mass on this side. So this would technically represent those two fetuses forming. And then in this process, we actually have a common placenta. So we have a common placenta and a common chorionic activity happening here in the middle. So in this example, there is splitting of the inner cell mass into two completely separated groups. The two embryos have a common placenta and a common chorionic sac, but they actually have separate, what we call amniotic activity. So if you look here, they share that orange placenta, and they share, or they don't actually have to share this activity here in the middle. So this is the amniotic activity would be this little clear circle that they're living in. So they're not technically like living in the same room. They're living in more of a 2-bedroom situation here rather than one, but they do share a common, what you could call a common home. So a common house, 2 different bedrooms is the way to look at it here. I guess in this situation with fraternal twins, you could look at 2 totally different houses. They're not even living under the same roof. And then if we're going to talk about identical twins, we have to go to part C right here. So part C, we have basically the exact same inner cell mass, and this inner cell mass actually splits. So this is the identical portion happening. So we have the splitting of the inner cell mass at a late stage in development, so it happens very far along. The embryos have a common placenta, so they share the same placenta. They have common amniotic activity, so they're living within the same room, basically, rather than a 2-bedroom. They're sharing that room. And they have common chorionic activity as well. So they're living in the exact same room, doing the exact same thing, sharing all their resources within the same area. And so once again, that late cell mass splitting apart, so happening even past this stage, that's what's giving us the identical twins. So A and B can be male or female. There can be 2 males, 2 females, 1 male, 1 female. They don't have to look alike. In this case, they don't even have to genetically resemble one another in any way. And then in part C, we have the identical twins who are going to be very genetically similar. And this just kind of breaks down the differences whenever we're talking about that variability in siblings. I want to be clear that twins do happen, and there's still variability even in the sets of twins that we see. So these are just a few errors that can happen in the process of mitosis. So if chromosomes actually fail to separate, abnormal gametes will form without 46 full chromosomes. So also you can have an extra copy of a chromosome, which we call trisomy, and that can result in genetic disorders. And as a reminder, meiosis occurs in sex cells, once again, and mitosis occurs in body cells. Okay, so here are the key takeaways for this slideshow. So there are 3 of them. The first being, in humans, meiosis generates haploid, what we call 1N gametes of both egg and sperm. The second is that crossover occurs during prophase 1, random alignment, and metaphase 1, and random fertilization. They all contribute to genetic variability. And then the third is that there are errors in meiosis that can lead to things like trisomy and other disorders. So this is the end of the slideshow. If you all have any questions, please follow up with me over email. If not, you all can move on to the next PowerPoint that I have. Thank you.